Hello everyone, welcome back to our channel. Today we are going to talk about influenza virus, that is commonly known as flu. The influenza virus is quite complex. So we will first talk about the structure. It has a segmented negative sense RNA genome with a capsid surrounded by an envelope. The genome consists of 8 RNA segments coding for 11 proteins. On the surface of the influenza virus, there are 2 major glycoproteins, HA and NA, which are essential for infecting the host cells. Inside the virion, the RNA segments are bound to the nucleoproteins and associated with RNA polymerase complexes. Now let's take a look on influenza virus genome. Influenza virus genome is composed of 8 segmented RNA strands. These segments encode 11 proteins, including the structural proteins and the non-structural proteins. The segmented nature of the genome allows for the genetic reassortments, contributing to the variability. These are some of the proteins that it encodes for. That is hemagglutinin, that is critical for binding to the host cell receptors and entry. NA is essential for the release of new virions from the infected cells. Nucleoproteins encapsulates the RNA genome. Matrix proteins M1 and M2 provides the structural integrity and facilitates viral assembly and release. There are some non-structural proteins, that is NS1 and NS2, that are involved in invading the host immune response and facilitating the viral application. So the replication cycle of influenza virus involves around the key concepts that are very basic for each virus, that is entry and attachment. The virus attaches to the salicylic acid receptors on the surface of the epithelial cells in the respiratory tract using hemagglutinin. And this binding triggers the endocytosis, allowing the virus to enter the host cell. After entering the host cell, the viral element fuses with the endosomal membrane, releasing the viral RNA segments into the cytoplasm. Then the viral RNA segments are transported to the nucleus, where they serve as template for the messenger RNA synthesis. And viral RNA polymerase complexes then synthesize the messenger RNA, which is exported to the cytoplasm for protein synthesis. Then the RNA segments are also replicated to produce new viral genomes. The viral proteins that are synthesized in the cytoplasm are then transported to the cell membrane. The new viral RNA segments are then packed with nucleoproteins and RNA polymerases complex, and the assembled virions are then bought off from the host cells, acquiring the portion of the host cell membrane containing HA and NA, and NA cleaves the salicylic acid residues, facilitating the release of the new virions. As we all know that the influenza virus is transmitted through the respiratory droplets when an infected person coughs or sneezes or even talks. So here are some common ways that it can be transmitted: through the breathing droplets, touching the surfaces that are previously contaminated with the virus, and then touching your face, and being close proximity to an infected person. We can prevent influenza by getting vaccinated and by practicing good hygiene, avoiding the close contact, using masks, and disinfecting the surface regularly to keep yourself safe. Thanks for watching.